Hi everyone, welcome to our Apply What You Know About Gene Expression video. And in this video, we're going to walk through a practice problem. You may see something similar like this on your assessments. And what I've given you is the template DNA strand of nucleotides. And in this case, I'm asking you to figure out the mRNA sequence that will be made at the end of transcription. Remember, that's also called the mRNA transcript. And then what will be the amino acid sequence made at the end of translation? And just a reminder, we can also refer to this as the polypeptide sequence. All right, so just to kind of orient ourselves, remember that DNA is a double-stranded molecule. I'm only giving you the template DNA strand, but from this strand, you could tell me the other strand of DNA, the one that runs 5' to 3', right? Because you know DNA, those two strands are anti-parallel and complementary from each other. Make sure to check out the structure of DNA video if you need a recap on that. Because messenger RNA is always made 5' to 3', we're going to go ahead and start there when we're writing out our messenger RNA. I probably should have written this a little closer so I don't get myself confused while I'm talking. So we know our mRNA is always made 5' to 3'. And so now to figure out the mRNA sequence, we're just going to use our base pairing. Because remember, the strands are always going to be anti-parallel and complementary to the template. So the first nucleotide with the template DNA is a thymine. So that's going to pair with adenine. The DNA base adenine is going to pair with the RNA base uracil. Make sure you don't put a thymine. RNA does not have thymine. Cytosine is going to pair with guanine. Okay, and I'm going to go ahead and finish. I recommend you pause the video though and write this part out on your own. All right, here we go. So then it's going to be C, G, U, A, U, U, C, U, G, G, U, A, U, A, A. Okay? So remember, it's just anti-parallel and complementary to that template DNA strand. Now, I do have this animated so it's a little cleaner and I can continue to write and it be kind of more obvious for you to see where I'm at. So I'm going to go ahead and just put that strand right back up in a little cleaner fashion. All right, so now remember what we need to do is we are going to have to read this messenger RNA in sets of 3 called codons. So here is our start codon, and it's always AUG. So if I'm asking you to build an RNA sequence that's going to be used for you to figure out the amino acid sequence, AUG will always come first, okay, because that's going to be the start codon. All right, so then we're going to do the next set of 3 and the next set of 3. And I'm visual, y'all. I've been doing this half my life, but I still need to underline them because it just helps me not get confused of where I'm at. All right, so now let's figure out which amino acid these codons code for. So AUG, so we're going to use the codon wheel here, and you start in the center and work your way out. So we have our A, then we go to this U, and then we go to, where is it? Here it is, G. Okay, and so that gives us methionine. Remember, you'll only need to put MET on your assessments. All right, next up, CGU. So we're going to start here. Always start in the middle. So we're going to go C. Remember, that's limiting us to this quadrant. So then now we're going to trace to the G and then to the U, and that gives us arginine. And again, I'm visual, so I just help keep myself on track. Let me go ahead and erase these doodles just so we can keep going. But while I do that, go ahead and be filling out the remaining amino acids. Oops, here we go. All right, AUU. So we're going to go AUU. That is isoleucine. And then we have CUG. That is going to be leucine. See, I was already getting confused because I didn't draw my arrows. Then we have GUA. So GUA is going to be valine. And then UAA is actually our stop codon. So there is no amino acid that's going to be delivered from this one. Remember, instead, a release factor would bind to the messenger RNA, and that tells the cell, hey, we're done. It causes the amino acid chain or the polypeptide chain to be released. The mRNA is released from the ribosome, and then the ribosome disassembles. Okay, so everything is released when we hit a stop codon. Okay. And so one thing I want to point out to y'all, and this is going to be really important for the next video when it comes to mutations, and I want you to notice, y'all, that the genetic code, right, our DNA, okay, so the genetic code, oops, I forgot a C, is redundant. What does that mean? Well, if you look here, let's trace out. Let's look at C, C, and then it doesn't matter what that third nucleotide is. We're going to get proline. So CCA, CCG, there we go, my pen went crazy. CCU, CCC, all of them give us the same amino acid of proline. This is why we say that the genetic code is redundant. Multiple codons can give us the same amino acid. And that's really important because the process of DNA replication, where DNA is made, isn't perfect. It's not flawless. It can make mistakes. That's where mutations come in. And sometimes mutations can happen without there being any serious consequence to the cell. Because let's say CCA was what the sequence should be. If that A got mutated and it was a G, U, or C, hey, we still get proline. Okay, so the fact that the genetic code is redundant can actually be very beneficial to organisms because it gives us a little leeway with some mutations, not all. All right, y'all. Um, let's actually go ahead and do the next slide in this video too, because it's going to build off what I was just talking about. It's going to be a good segue into our mutations conversation. All right, so for this prompt, it says a mutation changes one nucleotide in a bacterial gene. Explain why this mutation could affect the protein produced by the cell. Include DNA, RNA, and protein in your explanation. All right, so what I just showed you, right, is that in this case, if there is an error in the DNA, it may not impact the protein. And I actually need to go back and revise what I said because the sequence, the CCA, CCG, CCU, this is our RNA sequence. Okay, right? Because we're using the codon wheel. Notice there's U's in it. So we know this is RNA we're talking about. Mutations happen at the DNA level. But if a mutation happens at the third spot in a nucleotide, okay, in the third nucleotide in the sequence, then the RNA will also still be impacted, but it may not impact the protein that's made. And I'm going to clarify that on this next slide, but I just wanted to make sure I emphasized that what we're looking at, the codons on this codon wheel are RNA. Okay, so let's use another example, and I should have put this codon wheel on that slide. I'm going to do, let's pretend this is my DNA sequence and what it should have been. This, after transcription, will give me RNA. So it will be complementary, right? So think of base pairing. So my codon would be GCG, right? So let's go back and look at what that gives us. So GCG gives us the amino acid alanine. So that's going to give me alanine. Okay, and this is going to be I'm going to put AA for my amino acid. So that's what should be, okay? But now let's pretend that there was a mistake made during the DNA replication process, and this cytosine was not incorporated. A mistake was made, and instead, another guanine was added. Well, do we agree that if there's whatever the DNA is going to impact the RNA, right? So if we have a guanine here in the DNA, then when transcription happens, a cytosine is going to be included. Okay, well, let's see what happens to the amino acid. So GCC, hey, it's still going to be alanine. So we're okay, right? No harm, no foul in this example. And now we're going to talk a lot more about mutations in the next video, but this is just kind of a good little intro for it. And so let's take a look though, because I don't want you to think that mutations are no big deal, because they can be. Let's pretend instead that the mutation happens here at this first nucleotide. So instead of a cytosine, an error was made and an adenine was put into the DNA instead. Well, that's going to impact the RNA. So instead of what does A pair with in RNA? A U, right? So now we need to go to the codon wheel and say, okay, well, what is UCG going to give us? Oops, wrong way. Sorry, y'all. So UCG, that gives us serine. So we have now added or incorporated the wrong amino acid. And y'all, even one amino acid incorrect in a 500 amino acid protein can cause the protein to either not function properly or to not function at all. Because remember, proteins have a very specific 3D shape. If this change from alanine to serine causes the protein to fold incorrectly, it's no longer going to be able to do its job. Okay. So in this case, the protein may not function correctly. It may not function at all. And there are actually other types of mutations that we're going to talk about in the next video that have other consequences. All right. So stay tuned for that, y'all. And as always, please let me know if you have any questions. Bye, y'all.